next is the updates from the recent $1 Million Bike Ride Pilot Grant awardees. It is my pleasure to introduce Dr. Ed Kolodny, Research Professor of Neurology at NYU Grossman School of Medicine. Dr. Kolodny, the floor is yours.
Thank you, Calliope, for all of the organization and lovely work that you've been doing, keeping us on schedule. I want to acknowledge also the University of Pennsylvania Orphan Disease Center, which has been annually helping to raise funds for rare disease research through the $1 Million Bike Ride, to which many of the supporters of APBD Research Foundation have contributed their energies and their time. As a result, we have had the opportunity through the program at the University of Pennsylvania to award grant funds to several people working in APBD, including the two speakers that you'll be hearing from in a moment. I first want to introduce Wyatt Yu. Wyatt, of course, is professor of structural biology at the University of Newcastle on Tyne in England. And Wyatt, you did a wonderful job this morning as a moderator. I'm going to follow in your footsteps. Let's, though, enjoy now the fruits of your labors in APBD research.
Thanks, Edwin, very much for your kind introduction. And hello, good morning, good afternoon, everybody. It's a pleasure to be here once again to join the session. And I think you can now see the slides. So I'm going to put it into full screen mode now. So thank you very much for the opportunity to speak. Of course, thank you for the opportunity to be funded by this work, by the generosity of the scheme, as Edwin mentioned. So I recently relocated my lab to Newcastle University in the northeast of England from University of Oxford. So what I'm presenting today will be a little bit of work from Oxford on the inception of the project and the concept, and then paving the way to this funded project that will take place from now on in Newcastle. So Wyatt, I'm a trained structural biologist, and that's a very basic science if we think of it that way. And we're really interested, and I'm very interested in shapes of protein molecules. And there are many different proteins, and because they have different functions and they have different makeup, they all will have different shapes. Yeah? And the shapes of one protein can also change when it performs its function. And we think that understanding protein shapes is very important in trying to understand the function of that protein and to try to understand diseases that may be causing mutations to that protein, but importantly for sort of relevance to this section in therapeutic design. And we feel that shapes and what we call them as structures is a way to guide the design of new drugs, and particularly new small molecules targeting these proteins. And for over a decade, we have been studying the shapes and the structures of enzymes that synthesize glycogen. And in human, this involved 3 enzymes, and we have had an excellent introduction in the morning also already. The 3 enzymes include the first enzyme that primes the initial glucose chain initiation, called glycogenin, shown in green. The enzyme then, the next one that elongates this chain to extend it further in a linear manner, that's called glycogen synthase. We have heard quite a bit in the morning, colored in blue. And to finish up this process, they need to be branched and they need to be highly compacted. And that's performed by the branching enzyme shown in red. And over the years, we really have been trying to determine structures, and we have in our work include the structures of the first enzyme, glycogenin, and the third enzyme, branching enzyme. Yeah. And the structural information or the structural work on the middle enzyme, glycogen synthase, has always been evading us. Yeah. And as this morning we have heard briefly, and we are interested in this, and this is because this is of interest to the field of APBD and the audience here. And as we have heard, that there is a rationale for looking at this enzyme glycogen synthase, or we call it GYS1, because inhibition of glycogen synthesis by way of this enzyme could reduce intracellular glycogen accumulation and could serve as a therapeutic approach for multiple glycogen storage disorders, as we have heard in the morning session. Yeah. So therefore, we have been putting effort into understanding the shape of this GYS1 enzyme by way of initiating the process of drug discovery. And in short, to cut it, we have finally got there and have determined the structure of glycogen synthase, GYS1, using an approach called cryo-electron microscopy. Apologies for sort of having one specialist slide here, just to show that this is really quite a new technology, that we will look at protein samples that are vitrified and frozen into a grid like this. It will be put into a microscope. Sorry to disappoint you, it just looked like a box here. And then from this microscope, we will then be able to take thousands of micrographs. And each of these micrographs, like a picture shown here, will contain all of these little particles. And these are the particles of glycogen synthase. And by merging a lot of these micrographs, and also in each of them, thousands of particles, we will then get 100,000 of particles. And then if we group them together, if we average them together, we will be able to get the shape of glycogen synthase, and in a 3D manner, and in a resolution that is useful for understanding its function and design drugs. And this method is different from a method it's known called crystallography, because we use a different approach. I might add to a point there later. So this is the enzyme called glycogen synthase, GYS1, the muscle form. We have heard a bit this morning that it catalyzes the elongation of the glucose chain, and it uses a donor molecule called UDP glucose. And this enzyme is really a classical metabolic enzyme where it is highly regulated by phosphorylation that inhibits it, but it can also be activated by an activator molecule called glucose 6-phosphate. So the structure we have determined is of GYS1. For those who like to know a bit detail, it is GYS1 bound with a small piece of the glycogenin GYG1. Yeah. And I'm showing here very briefly the cartoon picture of how the structure looks like. It is containing 4 molecules of GYS1 together. I just put at the bottom that this is a piece of work that we have put into a preprint in bioRxiv, and the paper will be coming out shortly in a month or so. And I also want to give a shout out to another group in the UK who has also done very complementary work on the same enzyme and using the same method to also get to the same structure. So both papers are in the bioRxiv. And the data is actually already pre-released in the public domain. But for the purpose of this talk, I just want to show one more slide of sort of pretty pictures to show that the structure helps us to sort of zoom into the active site and then help us to understand how the molecule of UDP glucose is bound to the enzyme and allow the catalysis to take place. It also allows us to zoom into the regulatory site and to understand how this activator I mentioned called glucose 6-phosphate is bound there and how it performs its activating function. Yeah. And the main message here is that this approach called cryo-electron microscopy does give us to the degree of resolution that we can begin to use this to look at how are the drug molecules bind. Yeah, and I think that's going to be quite an approach in future and in upcoming years. And we, therefore, like other people, think that cryo-electron microscopy is rising to the stage in drug discovery and can complement another technique I mentioned called X-ray crystallography, which has been the main workhorse for determining structures or shapes of molecules. And we think that cryo-EM will be particularly useful for proteins that are quite big, quite dynamic, and also difficult to do crystallography, and primarily because the method of crystallography requires certain very stringent conditions. So we show here two examples from industry on other enzymes and using cryo-EM to show how molecules such as allosteric ligands or small molecule fragment screening molecules can be shown to bind to these enzymes, and using cryo-EM to understand how they bind and how they can use to launch drug development work. But of course, this approach of cryo-EM does have its limitations. For example, it is still quite low throughput, meaning it's quite a long process compared to crystallography. It is a very expensive setup, and it has limited accessibility in terms of places that have microscopes like this. And therefore, of course, a lot of industry groups will have sort of better traction in this approach than academic labs or colleagues like ourselves. But we do want to take advantage of the fact that we actually have determined the glycogen synthase structure by cryo-EM. And therefore, we want to think of a workflow where we can incorporate elements of cryo-EM, but also take other technology platforms to then look for small molecules, or in this case, inhibitors for GYS1. And so therefore, we decided to pursue two further technology platforms. And we are really grateful for the support of the APBD Research Foundation and the $1 Million Bike Ride Initiative from the Orphan Disease Center to allow us to pursue these two technologies. The first one is called the DNA Encoded Library, DEL. This allows us to screen hundreds of thousands, if not millions, of molecules in a test tube to look for those that bind to the GYS enzyme, for example. And each of these molecules to be screened contain a piece of DNA, shown here in the sort of helical shape. And the DNA serves as a barcode, so that you can then use a scanner, in this case, a particular reaction, to look for those that actually stick to the protein, therefore indicating that it does bind to the GYS protein. So that's one approach called DNA Encoded Library, DEL. The second technology we use is machine learning, a computational approach where we take all the hits, all the molecules that we identified from the screening using the DEL library. We analyze them together by computational approaches, and we try to use that data to then predict what other molecules or what new molecules can be used to bind to the enzyme, and to allow us to sort of make new compounds and then do the screening again. Yeah. So therefore, we try to do screening and then computational prediction, redesign some of these molecules, and screen again. And we believe that by doing this by several rounds, we'll get a lot of hits, and we will also improve the hits in each round. So this is my last slide. This is a project that we have just started in the past 2 months, thanks to the support of the foundation and from the scheme. And we have all the components ready in place. And I'm very excited that all the components of the DNA Encoded Library screening, the machine learning computational work, and some of our own cryo-EM or other biophysical work are all taking place in Newcastle. And it is a very unique setup in an academic setting that we can do. Yeah. And I've listed the colleagues here who will drive different parts of the work. And the $1 Million Bike Ride funding also is helping us and enable us to actually consolidate further support from within the university. And I'm glad to let you know that in the next 4 years, we will be provided a PhD student to continue the work in the next 4 years to really carry this process further along in the next few years. So I'm going to end here. Apologies, I'm not showing results yet, but I hope we will keep you updated, and we will share this in future meetings. So I'm just going to put on my last slide here to acknowledge my group in Newcastle, my former group in Oxford, and my alumni and colleagues. And thank you for everybody for your attention.
Wyatt, thank you so much. We're going to hold off on questions until after our next speaker, if that's all right. I'm going to ask now Felix Nitschke from UT Southwestern Medical Center, who is assistant professor in the departments of pediatrics and biochemistry, to update us on his current work and the results of his APBD research.
Thank you, Dr. Kolodny, for the kind introduction. Also, I would like to thank the APBD Foundation for the opportunity to talk at this conference, and of course for your fundraising efforts that also facilitate my research. The project I will talk about today I like to call Conquer from Within. In essence, I'm proposing to treat APBD by viral delivery of cross-correction-enabled amylase. Why amylase and what is cross-correction, I will explain shortly. As all of you know, in APBD, the glycogen branching enzyme, one of the central enzymes of glycogen metabolism, is mutated and insufficiently active. This leads to the formation of insoluble glycogen, which then accumulates in the form of polyglucosan bodies. Consequently, there are several treatment avenues for APBD. One is to downregulate glycogen synthase. Another is to replace the defective branching enzyme, and yet another is to remove the insoluble glycogen. In this talk, I'm starting off from the last one, to remove insoluble glycogen by delivering hydrolytic enzymes such as amylase. Now, we heard earlier from Dr. Gentry about an effective strategy to bring amylase into the cells of the brain. To make proteins enter cells, one way is to fuse them to fragments of the 3E10 mouse antibody. Proteins fused to these antibody fragments can enter cells that express the ubiquitous ENT2 receptor. For instance, the enzyme myotubularin was successfully delivered into muscle tissue by using single-chain FV fragments. As mentioned before, amylase was successfully introduced into mouse brains using the Fab fragment. These data from Dr. Gentry's paper show impressive efficacy in removing polyglucosan bodies in mouse models of Lafora disease. The protein that was used here was produced by secretion from cultured cells. The protein obviously could penetrate cells, and not only that, it was still functional and did not provoke an immune reaction. That made me think, could it be possible to utilize antibody enzyme fusion, such as Val0417, to advance gene replacement therapy in the brain? After all, Val0417 would be able to bring about something like cross-correction for cytosolic proteins in the brain. What is the significance of cytosolic cross-correction? I will explain that right now. Gene replacement therapy attempts to deliver a functional copy of the defective gene. The current challenge of viral gene therapy to the brain is that it only reaches up to 20% of the brain cells. Now, in APBD, insoluble glycogen accumulates in the cytosol of cells all over the central nervous system. So viral transduction of 20% of the brain cells with, for instance, an intact copy of the branching enzyme would likely not be enough. A way to overcome this transduction hurdle would be cross-correction. Cross-correction happens when a protein gets secreted from the cells where it is produced, distributed through the organ, and when it enters cells that are not producing the protein. This naturally happens to lysosome-targeted proteins, but not to proteins targeted to the cytosol. To cross-correction enable an APBD-relevant cytosolic protein, we essentially need two things: an efficient secretion signal and a cell-penetrating peptide. And exactly that is given in the Fab amylase Val0417. That's why I suggested to encode an antibody amylase fusion like Val0417 in an AAV9 virus and use that as a treatment for APBD. The advantages are that this approach would not need repeated administration, but with one injection, the majority of brain cells could be corrected. Here's an overview of my overall strategy. We have designed several protein constructs. Those have almost completed the phase of in vitro assessment and where we test secretion and cell penetration in cell culture. The most promising construct will be advanced to virus production and eventually used in mouse studies. At the same time, we're working on using this pipeline also for the delivery of different proteins such as GBE1. Now, a couple of words on construct design. Fab amylase, as it has been shown to work by Dr. Gentry, consists of two separate proteins, a fusion of amylase with a heavy chain fragment and the light chain. The complex is formed in cells that co-express both proteins. Now, to fit both proteins into one virus, this is one possible arrangement, embedded between a common upstream promoter and downstream enhancer elements. Both proteins will be connected through a so-called 2A self-cleaving peptide. This arrangement is called bicistronic, allowing both proteins to be expressed with just one promoter. Similar bicistronic viral therapy has been successfully used before in mouse models of Sandhoff disease with no adverse effects. What happens is that the 2A peptide sequence invokes something called ribosome skipping, which means protein synthesis will be stopped and restarted, and it effectively leaves only remnants of one or few amino acids in the upstream and downstream protein. As mentioned, both proteins have their own secretion signal, which have been shown effective by Valerian and Dr. Gentry. In addition, we designed other constructs that use a monocistronic arrangement where light and heavy chain fragments are connected to amylase and each other through linker sequences. This type of single-chain antibody fragment had been used successfully for the delivery of human myotubularium to a mouse model of X-linked myotubular myopathy. A potential advantage of the single-chain protein is its smaller size, which may improve virus packaging. A potential problem of this single-chain fusion protein is that secretion was not tested yet. For that reason, we designed a construct with an alternative secretion signal. One thing I should note here is that the bicistronic construct, after the fashion of Val 0417, uses an amino acid sequence that had been humanized. That means it differs from the original mouse antibody sequence in various places to make it less prone to provoke immune reactivity in humans. In addition, we developed single-chain constructs with a humanized antibody sequence. This allows us to keep the advantage of the humanized sequence and combine it with the small single-chain arrangement of the original mouse construct. The difference between the two single-chain humanized constructs is again the use of alternative secretion signals. We are evaluating these constructs now regarding their ability for secretion and cell penetration. All the while, we have been working on keeping the pipeline illustrated here versatile, so it can be readily adapted for the delivery of other cross-correction enabled proteins, including, for instance, the glycogen branching enzyme. Part of keeping this platform versatile was to include nanoluciferase technology for sensitive detection of secretion and cell penetration. For that, all our constructs contain a short 11 amino acid HYBID tag at their C-terminus. HYBID activates the inactive luciferase large bit, which results in the generation of a light signal, which is detectable with high sensitivity. We use this technology to confirm expression of our constructs in HEK cells. Instead of using primary and secondary antibodies, the Western blot membrane is probed with recombinant large bit protein to generate luminescent protein bands where HYBID is found on the membrane. We can also detect hybrids secreted into the medium of cells that were transfected with different amounts of our constructs. And in addition, the addition of a secretion inhibitor, Brifeldin A in this case, abolished the presence of hybrid protein in the medium. This confirms that the signal we see on the left side is generated by hybrid-tagged protein that was secreted from HEK cells. The nanoluciferase approach can also be used to detect hybrid-tagged proteins in live cells. For that, we generated an HEK cell line that stably expresses the large bit protein. Hybrid-tagged proteins in the culture medium that enter these cells will generate a light signal, and this allows us to quantify the degree of cell penetration for each of our constructs. To test our live cell detection system, we transfected varying amounts of a plasmid encoding a non-secreted form of amylase that carries a hybrid tag. The live cell luminescence strictly correlates with the amount of amylase expressed in the cell. For initial proof of cell penetration, we transfected HEK cells with a bicistronic humanized Fab construct and let the cells secrete Fab amylase for 18 hours. Then we transferred the culture medium onto large bit expressing cells, and after 6 hours of incubation, we measured live cell luminescence. We detected a significant increase in luminescence signal as compared to cells that were incubated with Fab amylase-free medium. Finally, we compared all our constructs regarding secretion. At first, we looked at secreted hybrid protein. Here it seemed the bicistronic construct secreted better than the rest. However, we also measured amylase activity in the medium. Here, the bicistronic construct was not performing as good. The reason for this is likely that the bicistronic construct produces two individual proteins, but only one of them has the hybrid tag. Most likely, the small light chain hybrid fusion was secreted much stronger than the larger heavy chain amylase fusion. To make the decision on which construct to advance to virus production, the amylase activity is certainly more informative. Interestingly, the secretion of the single-chain mouse antibody construct could be enhanced by using an alternative secretion tag. This was not the case for the humanized single-chain construct. In the following slide, I will show you our preliminary cell penetration results. We are currently completing these analyses to include the alternative secretion tags. However, we already see that cell penetration seems largely in line with the amount of amylase secreted to the medium. This indicates that all fusion proteins tested here are taken up in similar fashion, and that secretion efficiency will most likely determine the extent of eventual cross-correction. I'm coming back to our general strategy. Pending the final in vitro experiments, we have essentially identified the most promising construct for a first proof of concept experiment in APPD mice. We could show that our therapeutic proteins are indeed cross-correcting, however, until now only in cell culture. By selecting the best possible construct based on in vitro results, I'm optimistic that cross-correction of cytosol-targeted proteins will also be possible in APPD mice. That I would like to end my presentation, and of course not without acknowledging a few people who helped tremendously in the realization of this project. Thank you all for listening, and I'm happy to take any questions.